Next, we will hear from Professor Gemma Reguera. Dr. Reguera has a broad background in applied and environmental microbiology, with research projects spanning human, terrestrial, and anthropogenic environments. Her research is highly interdisciplinary and aims at harnessing microbial components, microorganisms, and microbiomes as technologies that prevent human exposure to contaminants, pollutants, and pathogens. She holds a PhD in microbiology from the University of Massachusetts Amherst and received a postdoctoral fellowship from Spain's Ministry of Science to work on the ecology of infectious diseases and metal cycling. Her group's research has also led to several patents and training initiatives to bridge the divide between academia and industry. She was elected to fellowship of the American Academy of Microbiology in 2019 and appointed in 2021 as editor-in-chief of Applied and Environmental Microbiology. Michigan State recognized her scholarship with selection as the William J. Beale Outstanding Professor in 2022. She is also known for her advocacy and initiatives to promote the full participation and advancement of women in the microbial sciences, efforts that were recognized in 2022 with the ASM Alice C. Evans Award from the American Society for Microbiology. She is also a great member of the College of Natural Science and helps out in lots of committees at the level of the college, and is just a great citizen. Please join me in welcoming Professor Gemma Reguera.
Thank you so very much, Dean Duxbury, for the kind words and introduction. I was just trying to crunch some numbers from what you said before. This is the 10th class with quizzes. I think I was part of the first one. So this is my 10-year anniversary. So I'm delighted to be here today. And I'm going to bring you to the world of microbes and the power that can be harnessed from those microbes. And disclaimer, I don't have a picture of Arnold Schwarzenegger. If I had known it was a trade, it was just going to be. Anyway, but I bring you these beautiful microbes that you see here, and I'm going to bring you electrifying discoveries that we have made thanks to wonderful people that I have had in my lab throughout the years. I wanted to start taking you on a trip to the environment, and not a pleasant environment. I think there is a lot of debate on how we're changing the environment, but we can all agree that we are changing the environment with our human activities, sometimes in very unpleasant ways. We usually focus on technologies, we focus on treatment of diseases, but we usually do not acknowledge how to treat the waste that links both processes as well. And one of the reasons why I became an environmental microbiologist was the motivation that even in these hospital environments that we are creating, microbes make a living and find a way to live. And they actually not only do that, they actually find ways to use those waste to grow and reproduce. And their activities are linked at the global level. So everything that we study in group of organisms has actually implications for our understanding of how we are changing and how we can reverse negative processes on Earth. And the question that drives our research is really this one that you cannot see because of the Zoom, but it is what can we learn from these microbes, the good microbes that we usually forget are the most abundant on Earth. So I want to introduce you to a really, really, really good microbe that my lab has been studying for the past over 15 years that I've been at Michigan State. It's called Geobacter. It's a group of bacteria. The name is Latin for bacteria from Earth. And this is already hinting at the fact that you can find them almost everywhere on Earth because they respire rust. Forget about oxygen. They actually respire this beautiful brown mineral that is just rust. It's a mineral oxidized form of iron. That's what it is. That's why you see the rust on nails when they get oxidized, for example. And there is a lot of iron oxides on Earth. Iron is one of the most abundant elements in our planet. These guys have found strategies to really capitalize on that abundance. And that's why they find them everywhere and they are so important. They are also the microbial miners of the world. They actually dissolve these minerals and a lot of metals of commercial importance that are sequestered in these minerals are actually solubilized so we can extract them. So there is interest in biomining as well. So this is the cartoon where you can see what they do. respiration is just discharge of electrons. We don't see it when we respire, but that's what is happening at the cellular level. This is what these microbes do. And their impact in all elements, cycle of all elements is huge. You can see that they make this iron soluble that we all need. That's why we get anemia when we get an iron deficiency. Any other living organism depends on the availability of this, but it influences the cycles of carbon, sulfur, and you name it. Every single intertwined cycle depends on this, and that's why their activities are so seminal to life on Earth. So let's focus a little bit on this one. When we looked under the microscope, what we found is that the cells that you see here, like these little gray sausages, rather than just being completely buried in the minerals so they can respire as much as possible, what they're actually doing is sending projections to actually get coated with these minerals. So this is the black iron oxide precipitates that are actually coating some filamentous extensions. So when you increase the magnification, you see that there is a very dense network of filaments that we found to be conductive. These are tiny, tiny little wires that the cells are able to make to discharge as many electrons as possible, which makes this process of mineral dissolution so effective. And I forgot to mention that there is another Latin word for you. We actually know what they are. They're called pili. They are actually very specialized pili. That's a Latin word for hairs. So they're just hair-like filaments, but there is something magnificent about them that allows them to function as nanowires. So which is the representation that you have in here. These are actually pictures of the cells with the animation to show you the electrical discharge. So they are electric microbes. And I have always found this fascinating. What can we learn from these microbes and what technologies can we develop to harness this power, which is what I want to bring to you today. So what I'm going to do is a little bit of unconventional, and I decided to actually tell you the story of the research in my lab, but I'm actually going to focus only on the stories that you can see here, but these are actually stories that made it into the news. And if you, as an alumni, you may have received magazines and you may have seen some of this work, so I'm giving you the whole background story. The first work that I want to tell you about is our finding that electric microbes can actually zap and immobilize uranium, which was something absolutely marvelous for us to discover. And this was efforts by one of my first graduate students, now Dr. Dina Kologi, that was so interested in this particular process and wanted to learn how we can use the microbes to actually immobilize uranium, a very toxic radionuclide. And the reaction is very simple. Uranium is actually this yellow solution. That's why it spreads so fast in aquatic environments and is so toxic because it becomes more bioavailable. So the idea is, can we zap it with these microbes and then the chemistry reverses and we mobilize it and make a mineral. So we have a form of bioremediation. And sure enough, she found that. This was years of work. What you see here is our little good microbe, the sausage. I colorized it in orange. And then this is the uranium precipitate, the mineral that they make. What you cannot see is that underneath is full of those wires that I show you. The wires are zapping it. And because we are microbial geneticists, we love to explore how much we can manipulate this. So we actually engineer microbes with more of the wires, which I am represented here. We actually make one that was a couple of strains that were so useless because they didn't have the wires and they died because the uranium got in. And then everything became mineralized from within, and that's how they die. But then we made this super bug that was actually so hairy, and it loved it. It zapped as much uranium as you can imagine, and it survived just fine, very high concentrations, toxic concentrations of uraniums. So what we did after this reaction is, get a nice paper, and then the news came out, and I think it was a little bombastic. Microbes generate electricity while cleaning up nuclear waste. It was a little bit of an exaggeration, but there was a lot of truth, and we built up on these discoveries to actually get as close as possible to this. But the important thing is that we did have a technology in which you could go to the environment contaminated with uranium. We know we can find some of these microbes because they survive that toxicity. We can add their favorite food, which happens to be vinegar, acetate, and they grow. And we can actually, at least transiently, mineralize the uranium that is spreading from the plumes of contamination. So that's the whole story about it. So there is a lot of potential for us to actually find ways to really use these microbes, stimulate them in situ, add them. We grow them in mass in the environment, our super strains with more wires, and really find solutions for the nuclear waste that has been stored since the Cold War and is leaking and contaminating the environment. And this made it into the press. It was huge. We love when we get these photo shoots because we get to wear lab coats we don't wear them very often. So here it is. Dina Sanella was a postdoc that participated in the work, and Allison, another of my first year students. And one of the surprises is that we started to get articles that said all-female team trains microbes to clean up nuclear waste. And it shocked us that they pointed at the fact that we were all females because we had not realized that we were all women. I mean, we knew we were women, but that it was anything unusual. We were scientists and we were working together as a team. But to me, it was an epiphany and a realization very early on as an assistant professor that there was tremendous power as a role model and a mentor in STEM. If we want to really diversify fields like ours that are still dominated by men, and if we want to really encourage women to show their talents and really capitalize on that power, that workforce power that we have. So there is a risk and reward, and that's why I like to just mention a little bit outside of research. We have high expectations of women and minorities to do the work, and this is a teamwork where we should all collaborate. And I like to bring up, this is a tweet from a few years ago from Professor Dame Carol Robinson, a very famous chemist from the UK that has been creating a lot of awareness and initiatives about how we have to support women in science. What she said is that if I speak out about women's issues, people think that my science isn't going well. I have heard that about me as well, and that I need an excuse to talk about something else not related to my work. And it's not that we like to hear our voices, it's that we were given an opportunity that is a risk reward, it's an investment, but a necessary investment, and that's why we took the opportunity. So I just want you to think about it. You're going to see a lot of women in the research that I'm going to showcase for you afterwards. So with that little break, just let's show how much power these talented young women had in my lab. Dina actually took the project to the next level and found that we could boost this armor by stacking cells on surfaces and materials with those wires and making these communities that were so resilient and so efficient at zapping that uranium and making the mineral. What you see here is a lot of the uranium mineral, all these kind of sheet-like structures like flowers. They are the uranium mineral and the cells underneath working as a team to make the process more efficient. And then it took us 10 years to, this is one of my current students, Morgan Clark, to really learn about a mystery that we had, and is the fact that we could account for 75% of the uranium that the cells were actually mobilizing based on the nanowires. That's the 75% here that you see here. But we could not account for 25% that was not mineral, but was absorbed by the cell. And that was a lot of uranium for the cell to survive. So there had to be some way in which the cells, the orange part, was actually maybe protecting itself and capturing, sequestering that uranium. So what Morgan discovered was actually that the cells are coated by a special material, a lipopolysaccharide, that is like a sponge for that uranium. And that's how it prevents the uranium from getting in the cell while the nanowires are doing their work. And this is very interesting because once it gets saturated, it actually sheds little vesicles. It remodels its membrane to get rid of that waste in little vesicles, which are these little blobs that you see in here. To the point that Morgan found that we can actually knock out the nanowires, and the cells, just having these mechanisms, manage to survive growing in the presence of uranium as well. And this is great because now we are studying these vesicles. We want to scale up the process and maybe use them as decoys to detoxify environments, to very rapidly sequester the uranium, at least transiently. And of course, this made it into the news, and then it's bacteria clean up radioactive waste. They don't like radioactivity, but they definitely help us clean up the uranium that is most abundant, the isotope that is used for enrichment for nuclear waste. That's the one that is not radioactive, and these microbes can actually help us actually clean up. And then my postdoc, Sanela, managed to isolate these nanowires, and then we made it again into the news. This was 2016. They leave man-made technologies in their dust. That was beautiful, I don't know, but they are really, really interesting. We were able to isolate them. They are all made of protein, which give us the ability to engineer them genetically. And then Sanela did beautiful work actually looking at the conductivity and learning exactly where we can apply these nanowires. What are the places in electronics where we have opportunities for this type of bio-based materials as well, which we can mass produce. And this is the work that I'm going to tell you, a teamwork by 3 wonderful students that have now graduated. Becky was a fantastic molecular geneticist that was able to look at models that we built of those wires, identify that they are made of a single building block, a peptide, which is made of amino acids. So we had absolute control of those little bricks to make the wires. She engineered them, and she created platforms used for fermentation at the industrial scale to actually make and mass produce these peptides. So we had the bricks. Angelina is a chemical engineer, and then what she did was scale up the process. So we had the peptide building blocks, and then she was able to scale up in chemostats the production, and she was able to develop the first approaches to assemble them in tiny little wires. And then Krista did the magic to actually standardize the process, increase the length, so we have tunability in length, which is very important for applications in electronics, and mass produce them. So we have a ton of nanowires that the cells by themselves, the native cells, could not do as well. And this is great for industrial applications. And now our work really is moving into green electronics. And something else that we found with these studies is that we figured out how these nanowires were so good at binding the uranium. In fact, what you see here in this color picture are actually charge distribution. Where you see red is negative charges, where you see blue is positive charges. And there is these tiny little hooks that are perfect to attract uranium, which is positively charged. And this is a model where you see the uranium. But in principle, we could bind and zap many other metals that are also positively charged. And this is something that we are exploring right now in my lab. Marcella and Hunter are students now in my lab, graduate students, and what they decided to look is at using these wires to bind cobalt. And they found that we could also zap cobalt. For microbiologists, this is a fantastic reaction because cobalt was believed to be too toxic to be mineralized by bacteria. And it's also at the thermodynamic edge. The bacteria are really not growing as much with it, because what happens is that they're just detoxifying it. It's a selfish mechanism. But when we made it into the news, it was great because I was trying to explain to the journalists that these guys are like Iron Man. When they put the suit on, when he puts the suit on, becomes so powerful, they just have to zap some of the cobalt and they become metallized like a super powerful armor. So they can actually keep removing more and more cobalt and actually eventually gain energy for growth. And we became very interested in this because cobalt is a very critical element to use for electric vehicle batteries. And what you can see, this is 41% of the use of cobalt just for batteries, superalloys for space exploration and applications require cobalt. And we have a big problem in the US currently because almost all the cobalt that is mined in the world comes from this tiny little region in Africa called the Copper Belt. And then the problem is that not only mining is now in the hands of foreign interests in this region, but the processing is almost always concentrated in China. So there are tremendous issues now about how our economy and our national defense is highly dependent on the supply chain for just this tiny little metal like cobalt, but then others as well. So I've become very interested in circular economy, and I'm part of a grassroots movement here by faculty, mostly economists, engineers, and sociologists on campus are trying to develop a circular economy institute. And I think just 2 weeks ago, we presented our pitch presentation to the provost as one part of the Great Ideas Summit. And we are moving forward on our own because we see tremendous value for the state of Michigan in particular to be able to address supply chain problems, that we have technologies to apply and to really circularize the economy, especially when we think about the electrification of the automobile industry in the state of Michigan, for example. And what we are doing is we have technologies for, think about cobalt, for electric vehicle batteries, but what we don't have is management processes for getting the residual waste treated and bringing all the raw materials back into that circular economy. And my group is actually working on technologies, and we're working now with electronic waste recyclers in Michigan that are starting to identify processes to recycle electronic waste with this target as well. So keep us looking forward. We hope to have an institute developed for this very soon and make Michigan State really a leading institution in circular economic issues. I told you a lot of things about the environment, electric microbes. So the next part of my talk is actually going to give you a hint of this power that was in my talk, and you may want to see some electricity, I imagine, rather than looking at metals becoming minerals. So really, what are the real applications? How can we harness that power, the electricity that we hinted? So I'm going to tell you a story that was started by my first graduate student, Dr. Alison Spears, in which she actually found ways to develop fuel cells with microbes, powered by microbes, which is what you see here. If you're familiar with fuel cells, it's a two-compartment type of reaction where the electrons come from this chamber, they create electricity, and then you have reactions here to capture those electrons and protons. Every time you move an electron, you have to move a proton, actually go through this permeable membrane or electrolyte, for example. So we replicated something like that in this tiny vessel so we could study and optimize the process. What we found is that we could actually attach the cells and with their wires, which you can see them here as an electrifying network on electrodes. So we could actually power electricity from here and collect it here. And actually we did experiments to actually make hydrogen over here. So we had a perfect harvesting of those electrons. What was more important than the electricity was the fact that we could add waste. That was the technological value. We could process a lot of waste from fermentations in these units. And the value of this, I can't really emphasize how important it is to go for the waste. This is something that was captured in the first news press release. Microbes provide solution to energy issues. We envision decentralized solutions. This is one platform that we developed. We use corn stover, that is a waste product in agriculture. It's treated chemically with a process that was developed and it really scaled up here by Bruce Dale here at Michigan State. And this is a proof of concept experiment. We made bioethanol, but all the waste were removed immediately by a bioelectrode that we developed for this application. And this was great because this reaction didn't have waste accumulated, so it was going really at high rate and very efficient. It became very important internationally. That's why our patents sometimes also become protected internationally. And we envision this as decentralized solutions for waste treatment and power generation. There are many places in the world where this type of technologies are seen as very, very important to address energy issues. And this became one of the top methods of turning waste into energy in 2019 in this press. We develop other platforms. We can treat glycerol and ethanol. We've become more sophisticated. We can mix different microbes in the bioelectrodes. We can engineer different activities in the bioelectrode. This is very important to treat the glycerin waste of biodiesel companies, and we work with the only biodiesel company in the state of Michigan to really test these technologies. And we were able to convert the most crude glycerin that comes out of the biodiesel factory into bioethanol, but also other chemical precursors of industrial importance that actually create more profitability for biodiesel companies as well. And of course, we patented these technologies. And this is one of the news clips. Patented bioelectrodes have electrifying taste for waste. And this is really the key, the waste. And what we were doing is really thinking about how we can scale up these processes. And that's very important to move from the lab to industry. And to do that, we were very lucky to actually have funds to commercialize the technologies and scale them up. And a postdoc, Bhushan Awate, was instrumental to this. He was a chemical engineer. He developed different reactors in which we could put our bioelectrodes. These are just some lab scale reactors that you can see. And then with funds from the MTRAC, you know, Michigan State is a state hub for ag bio research and commercialization of ag biotechnologies. And it takes time and investment to really see how we can bring those technologies from the lab to real applications. And we started with an idea and a fuel cell that is not scalable. And thanks to Bhushan, we were able in 3 years to move to an optimized platform that was profitable for the treatment of glycerin to industrial chemical precursors that biodiesel companies could actually make most profit of. So what we have now is the patents, hoping to license, move into pilot scale, and then hopefully commercial applications as well in the long term. And this is what my mother says. It's a picture where I have my dad's murdering eyes. That's why I'm showing it to you. They wanted to make it. It was for the campaign Spartan's wheels. Apparently, this is a very willful look. And this is where the tiny innovations that we're making with microbes can actually have big impacts to highlight all this research. And I was very fortunate to be selected at the Innovator of the Year award. But my head, you know, my face is the visible one, but this is something that wouldn't have been really achievable without the tremendous contributions from Alison and Bhushan throughout the years. So what Bhushan did is I asked him, okay, we need to show that we have power in these reactors. And then he did this, can you turn it off for a second? Yeah. He actually, I asked him, can you put lights, like Christmas lights, something really not very glamorous. But he was very quiet and I didn't hear from him for weeks. And when he came back, he said, I have this installation. And he had the video. What he did is he went to the MSU store and bought the Spartan fan, which is powered by 3 AAA batteries. And then he actually plugged it into the reactor. And what you see is the power that we're getting into just a small half-a-liter reactor. So this is the power that is coming from just a very small unit because we're just short-circuiting to see it, and it's just removing the waste. It's a failed experiment because it was more power than we anticipated, so we couldn't actually see the message on the fan. So these are the screenshots to show you go green and a Spartan wheel. So this was disappointed, and I wanted to actually make the experiment work. So I'm going to show you another video. This is part of there is a story between this, the Bill and Melinda Gates Foundation and the Electrochemical Society, who has always been so welcoming of me and my research. They actually invited us to Cancun to a conference to actually identify solutions for sanitation. And between margaritas, which I love, they asked us to send proposals to address sanitation issues in the world. And I must have been very inspired because they selected my proposal to use these powerful reactors for sanitation. And not only that, to actually remove human urine. And I actually made it to the Shark Tank presentation and I got it. I was actually traveling back to Michigan when I got the message that I was one of the awardees of this Gates Foundation awards. And what I promised, and then on the plane, I had to figure out how am I going to actually make it work. I told them that I could actually get human urine to make ethanol to sanitize like toilets and then create clean water and electricity maybe to flush. And there is an idea, maybe the margaritas enlightened me, human urine is actually sterile, also is great. And then it actually, if you look at the chemical composition, is perfect for bacteria to grow. It has a lot of nitrogens. If you add a waste like a carbon source, potentially we should be able to do it. The ethanol is the way to disinfect, so nothing grows in it. And then you can recycle that water. And you may have heard that there are many areas of the world that are starting to have diverting toilets, because the problem is the poop, as always. You don't want the urine to be mixed with the poop. So that was the whole rationale and all the inspiration that I got in Cancun. So I told Bouchang, poor Bouchang, can we do it? And you can see the yellow part. It took us forever to get permits because nobody at MSU knew how to treat urine and what permits to get. So they told us, as long as you produce it yourself and you don't collect it from volunteers, you're okay. So Boucham volunteered for science and brought monthly production to the lab. The lab members complained about the smell, but what I'm going to show you is a reactor in which we are actually doing exactly this. And you can see that it finally worked. Michigan State, go green. And thank you all so very much.